Hi, so this is Allison. I am in the tissue culture room. I'm going to be passaging 2 flasks of our 3T3 cells, and I will be posting explanations as we go. So as always, we should gown up, which you have your goggles first, and then your gown. So the key when it comes to putting on PPE is to put on the least contaminated to most contaminated piece of PPE in that order. And then when we remove our PPE, we will do the opposite order, which is why we usually take off our gloves first. After we have everything on, we are going to give our hands a little spray of ethanol just to keep everything really sterilized. So earlier, Katie and I put some of the growth media into the bead bath, and this warms up the media to around 37 degrees Celsius. If they are still at 4C when we passage them, these cells will not be as happy. So this is the microscope that I will be using to check the cells. I checked them earlier, and they were very, very confluent. If they're too confluent, as in when there are too many cells, then they start getting a little fussy. So that's why we are going to passage them today. And I have turned on the Biosafety cabinet so then it could run a little bit, and I'm going to spray everything down with ethanol. Okay. Perfect. So all of the air is actually sterilized. The safety cabinet sucks the air in through these ducts on the bottom, and then it goes up into the system, and then it gets filtered, and then it comes back in. So it kind of like cycles through, which is good because then any contaminated air will be filtered out. However, if there are dangerous chemicals, for example, you'd want to use a fume hood if you're working with toxic chemicals. But for the TC room, a Biosafety cabinet is perfect. So we're going to spray everything down again. Put that in. This is very important because mammalian cells, such as 3T3 cells, are very susceptible. If we get any kind of contamination, it could cause a lot of issues for the lab. We're going to passage one with antibiotics. We're using pen strep, as you can see here. It's going to focus pen strep. And we're also going to passage with regular media, which is right here, 3T3 media. And of course, we have our trypsin. So trypsin is an enzyme. It cleaves all of the adherence proteins, and this is why it's really good for lifting cells. This is PBS. It is a buffer solution that we use to rinse off the cells. So this is a special TC room incubator. It holds the heat at 37 degrees Celsius, and there is 5% CO2 at all times inside of the incubator. So as you can see, there's a double door. We have to do this very quickly. As you may have noticed, there are 2 doors. It's important that you don't breathe too much when you open the second door, or else you might contaminate the inside of the incubator. So here we have Carol and Amanda. is what these cells are called. As you can see, like, the solution is a little bit more on the orangey side. This means that there has been a lot of cell activity. In the way that the growth media has phenol red, which is a pH indicator, and when the environment is acidic, the yellower the media becomes. But these cells are really, really, really confluent. They're more than 90% probably. It is perfect for splitting. Let's see. Okay. Make sure when you spray the flasks, you don't spray here, because this is where the air can go in and out and it could be bad for the cells. So you really should just spray around like this, down a little bit. And I will determine how many flasks I want to split this into after I see what the cell count is. Because generally we do 500,000 cells per flask. These are the serological pipettes that we are going to use to measure everything. Spray them down. In the biosafety cabinet, we want to make sure that our hands don't cover this area, because that's where the air goes in. When we aspirate, we have these pasture pipettes, I think is what they're called. They've been autoclaved, which means that they've been heated to a high enough temperature that would kill most contaminants. We are going to just do this. Aspirate it. So when you aspirate, you want to make sure that you have all the old DMEM on the other side of the flask so you don't hurt any of the cells. So after that, this goes into the sharps container. So next we're going to rinse the cells with PBS. So I'm going to put 5 mils of PBS in both containers. And since I don't want to contaminate, I'm going to use different pipettes each time. Alright, so now that we have the PBS in, gently rock the flask so then it can basically rinse off all of the cell debris, and after this we can turn the aspirator back on. So we want to add 2 mils of trypsin per flask, and I have to do this fast because we only incubate the trypsin for 2 minutes in these flasks, because if we do it for too long, these cells might die, and that is not good. So the next step, I'm going to put these back into the incubator for 2 minutes, and while that is happening, we are going to set up the cell counter. Yellow. Okay, the camera died, but that's okay. Oh, hello, Jeannie! I'm filming on my phone right now. You can say hi if you'd like. Hi! When you incubate with trypsin, after the allotted time, you will check on the microscope again to see if there are any more adhered cells. So earlier with this flask, there were some more adhered cells when I checked on the microscope. So I put it back into the incubator for a little bit longer, just to let the cells have time to dislodge. We're gonna go to the next step, which is to add growth serum or growth media, also known as Dolbeco's modified eagle media, DMEM, which is the most common kind used in tissue culture as far as I know. I already added 10 mil of DMEM into this flask. This dilutes the trypsin and prevents further cleaving of the proteins. So right now I'm just pipetting up and down to break up any clumps, as well as doing this waterfall motion, because the step after this is cell counting, and we will see what concentration of cells we have, and we want to make sure that we have the right amount of cells in each flask. So first I am going to put 90 microliters of cell solution into the Eppendorf tube. So we are going to pipette 10 microliters of trypan blue into each of these cell solutions to dye the cells. And then here we have the cell solution, and then we just add it in, and then we're gonna pipette up and down to dye the cells like that. This is a hemocytometer set. We have this piece of this like glass piece. There are little grids on the glass. We use these grids to count the cell amount. So what happens is that we calculate the average number of cells in these 4 quadrants that show up when we look at the hemocytometer under the microscope. And that will tell us, after you multiply that number by 10,000, will tell us the number of cells per milliliter. We're going to put the glass cover on top of the hemocytometer such that it lines up with the edge. And we are going to pipette 10 microliters of solution into that divot, so then it'll spread out the cells. We are going to take out 10 microliters of cell solution, and then we're just going to pipette it into the hemocytometer, and then ta-da, that is done. Okay, never mind. For some reason the camera's not focusing today, but I have gone ahead and counted all of the cells.
Rocking back and forth like you rock the flat.